Hi guys, so I want to talk about an interesting topic that's going to kind of revisit something we addressed before about normality. You recall that many of our tests that we do are parametric statistics, and they therefore assume the normality of our distribution. So for example, when we developed our special t-test, we assumed a normal distribution around mu, and we estimated the standard error around that mean from the sample data, and we would ask, for example, what was the probability of getting a y bar like that, that far away from the hypothesized mean. And we had to assume a normal distribution in order to do our simple t-test that we performed to come up with a p-value associated with that probability of getting a y bar like that. Okay, so and I said that we had to assume a normal distribution. Now, if we start to advance to tests in which there's more than one group, more than one sample, we, for example, if we had two groups like this, and we had a y bar for group A and a y bar for group B, there are a couple of assumptions that we're going to have to make, and that is that the data are normally distributed again. And we're going to have also the assumption of homogeneity of the variances. And I'll get to that one a little bit later. That is, the spread around those means is the same. But we're also going to still be assuming a normal distribution. And so I want to focus on this one for now. And I want to pose to you a puzzle. So let's look at the distribution of the data. Now, I've plotted them like this as two curves. But if we were to actually just plot the data and the frequency of those observations, what it would look like would be this, right? In other words, we have a stack of data here, and they overlap, but we have two peaks like that. And that's clearly not a normal distribution, okay? So that's not normal. And yet, so far, I've just talked about a test, Shapiro-Wilk-W test of normality of the data. So it's very important that we understand that it's not normality of the data that we're interested in, because the data in this case look like this. Instead, it's the normality of the deviations from these means. Okay, so it's the normality of each of these curves. And what's being normally distributed here is the deviation from the mean. So what do I mean by that? So the deviation would be, so here's our y bar for group A. All right, we have observations that go into this, and if we were just to plot them, of course, there would be no height to this graph. It would just get less frequent the farther out we go, and there'd be lots of them in here. But if we plot those as frequencies and kind of make classes, okay, so we get a nice humped curve, a normal distribution. Okay, what is a residual? So what we need to do is kind of look at one of these points. So here's an observation, y sub I. Individual I in this group that I've sampled deviates from the mean, right? And in fact, I could just subtract yi minus y bar A, and that would be the deviation of observation I from the mean, y bar A. And that deviation is the residual. Okay, so the residual can generally be written as y sub I, the value for a given observation, minus y bar for the group, so this is the group to which I belongs. That way, if I have a second group over here, and I have y bar B, and I have an observation from population B, I don't subtract from y bar A, I subtract from y bar B. So here I would get observation J minus y bar B. And that would be the residual, okay? So either of these are residuals, and in fact, when we pool all those residuals, what would the mean of that be, by the way? So if I plot the residuals, all right, you figure it out? Mean is 0, right? Because we're looking at deviation. So some will be above, some will be below. On average, they'll be 0. And if we pile all those residuals on top of one another, that's what we need to test the normality of. Okay? So that is expected to be normal, but obviously this is not normal. The data are not normal because we have a 2-hump distribution here. If we have 2 groups. If we had 3 groups, we'd have 3 humps, potentially, if they were separated means, right? Okay. So we do our Shapiro-Wilk W test on the residuals. And I'm going to show you in class how to get the residuals, but basically in SAS Jump, what we do is we, in the process of setting up the model, you'll see once we've set it up, we run the model, we are going to save the residuals. Before we even look at the results of the test, we're going to save residuals and create a column in the data set, which is all the residuals. And once we've done that, we're going to actually test the normality of those residuals using the Shapiro-Wilk W test. And that's an iterative process. So we might, for example, find that the residuals deviate from normality. We'll do a transformation. We'll run the model again. We'll save the residuals. We'll test the normality of the residuals. And this is a repeating process that goes on until we've got normality of the residuals. Then we can go ahead and perform our test on the transform data. All right, now I want to point out one thing. There are residuals for any continuous y variable, whether we're doing regression or whether we're doing ANOVA. So here's showing a regression, for example, and we have deviations of observations from a regression line. Well, those are residuals, and you'll see some of them are negative, some of them are positive. The mean of them is going to be 0 if we've optimally fit that curve, right? And so we have an assumption with regression of normality of residuals around the regression line. And so it doesn't matter whether we're doing an ANOVA, where we're comparing groups that have different means, for example, and we have groups A, B, and C. We assume normality of residuals. Now, I do want to talk to you about how we can run into problems sometimes, and that is that the higher the mean, the broader the range of values, okay? And so you can get large means and small means having very little variation, large means having large variation. That doesn't violate the assumption of normality necessarily. Those could still be normally distributed around the mean, but there's a separate problem there, and that's heterogeneity of variances. And I want to point out just now, while we're talking about this, that regressions and ANOVAs assume homogeneity of variances. So we'll have a separate test where we look at that issue. All right, so that is what a residual is, and you're going to be looking at residuals a lot when you test for normality.